So, hello, good morning, good evening, good afternoon, good night, whatever. Today I'll be revising DADH. Again, yes, again DADH. So, आज थोड़ा सा हाँ आगे का पार्ट करेंगे कल जो किए थे उससे. This is just a general introduction to histology. Now, today, bone histology of bone. Okay, चलिए. Now, there are three types of cells in bone. Let me sit properly. Yes. Now, three types of cells. One is osteoblast, osteocyte, osteoclast. Now, what do you mean by blast? ये वो वाला blast नहीं है. This is forming. Blast means forming. Clast means destroying. And site is just बाद में. Now, osteoblasts are bone forming cells. Now, some of the cells get entrapped in the matrix of the bone, in the bone matrix, and hence they shrink and are called osteocyte. Now, some of the osteoblast cells, they get trapped inside the bone matrix and they shrink because they are trapped, they shrink and are now known as osteocyte. Okay. Now, osteoblast पढ़ लिया, osteocyte जान गए. osteoclast bone cells helping in bone degradation, destruction. Okay. Now, how do you study a bone which you have taken from anyone's body? और मतलब वैसा नहीं, but ठीक है. Preparing. This is practical stuff for study of bone. Now, there are two methods. Tongue twister. One is demineralization. Another is ground section. Now, in demineralization, all of the now minerals means inorganic कंटेंट. अब डीमिनरलाइज करेंगे तो इनऑर्गेनिक कंटेंट हट जाएगा. तो डीमिनरलाइजेशन किसके लिए होता है? टू स्टडी ऑर्गेनिक कंटेंट. क्योंकि डीमिनरलाइज कर दिए, इनऑर्गेनिक कंटेंट निकल गया तो ये दिस इज ओनली फॉर ऑर्गेनिक कंटेंट. ठीक. ना ग्राउंड सेक्शन जो होता है इससे सब पढ़ सकते हैं. ठीक है. In demineralization, they use acids and some chemicals to uh, remove all the inorganic content, and what is left you cut thin sections of it and study under the microscope. And ground section, for example, if this is a bone, you just keep on grinding the bone until you're left with a thin slice of bone. इतना पतला, ऐसे करके ऐसे, मतलब ऐसा करके ऐसा पूरा पतला सेक्शन, इतना पतला कि वो लाइट भी ऑलमोस्ट पास कर रहा हो, तभी तो पढ़ पाएंगे. सो दिस इज ग्राउंड सेक्शन. यू जस्ट कीप ऑन rubbing it with something until you get a thin section. Now the covering outside and inside the bone is known as periosteum. It has two layers. Now outer fibrous layer and inner cellular layer. Periosteum has two layers, outer fibrous and inner cellular. Now in X-ray, if something shows up as black, it is called radiolucent. Something shows up as white, it is known as radio-opaque. Radio-opaque means highly mineralized. All the organic content will generally show up as radiolucent, means radio waves will X-ray can pass through it through organic content, but through inorganic content like minerals etc. the X-ray cannot pass through, है ना. तो इसलिए bones are generally seen white in X-rays. Now something about WBCs. इसी में पढ़ेंगे. Yes, WBC. Now WBC, there are two types: agranulocytes and granulocytes. NEB. ग्रैन्युलोसाइट्स का 3 होता है. न्यूट्रोफिल, ईओसिनोफिल, बेसोफिल, लिम्फोसाइट्स, मोनोसाइट्स. ग्रैनुलोसाइट्स का NEB, न्यूट्रोफिल, ईओसिनोफिल, बेसोफिल. A granulocytes, monocyte and lymphocyte. Now according to size, largest to smallest, NLMEB. Neutrophils, lymphocytes, monocytes, eosinophil, basophil. Size होता है, number होता है. No, not size. number. Hmm. Say this. Yes, number or percentage. Now, let's discuss neutrophils. Now, neutrophils are I think, the largest and have both acidic and basic granules. Since न्यूट्रो मीन्स न्यूट्रल, तो एसिड और बेस दोनों होगा तो न्यूट्रल हो जाएगा, है ना. तो let's say these are basic acidic granules and these are acidic granules. So no, basic, ठीक है, जो भी, and their nucleus has 3 to 5 lobes. अब यहाँ 2, 3, 4, 5, and nucleus is not in a मतलब not in, कैसे बोले इसको, एक ही साथ नहीं है, not like in a globule. It is elongated, है ना, and it can be in any shape. Circular globules lobes होते हैं, but it is elongated. Now neutrophils, eosinophil, हिट्स नहीं, PHILS, T नहीं है भाई, इधर नहीं दिख रहा है, मतलब नहीं है. Eosinophils. Now eosinophils have dumbbell shaped nucleus. dumbbell नहीं, earphone, headphone. Yes. Acidic and coarse granules means acidic granules हैं and coarse granules which are, ठीक है इतना ही, and nucleus has 2 lobes. Neutrophils, eosinophils, basophils. Now basophils have a kidney shaped nucleus. And have basic coarse granules. Nucleus is kidney shaped and NEB lymphocytes. They are of two types, small and large. Small has greater nuclear cytoplasmic ratio, higher as compared to large. Now monocytes. मोनोसाइट क्या होता है भाई? मोनोसाइट्स लेट मी चेक, मैं लिखा नहीं हूँ। सो मैं जिस कलर बता दिया, बेसोफिल का सा किडनी सेक नहीं होता है। बेसोफिल का सेम ईयरफोन शेप्ड होता है, बट इट इज थिकर। किडनी शेप नहीं होता है, ठीक है। एंड इट हैज बेसिक ग्रैन्यूल्स। मोनोसाइट का किडनी शेप न्यूक्लियस होता है, एंड दे आर जनरली लार्जर। अब लार्ज तो मैं बनाया नहीं, चलो लार्ज बना लेते हैं। ठीक, ओके। नाउ नाइस। Now something about स्टेन्स। ये सारा प्रैक्टिकल का है। स्टेन्स, H and E stain, hematoxylin and eosin. Hematoxylin is for cytoplasm. Eosin is for nucleus. This stains the cytoplasm shades of pink, and the nucleus is shaded bluish black. Van Gieson stain, VGS. This is for mainly to identify collagen fiber, which is stained red. Now let me show कि कैसा होता है VGS। Now this दिख रहा है, हाँ, this is Van Gieson stain, See, collagen fiber is red and all the nucleus will be black, bluish black. Collagen fibers are red, cells are yellowish green, and the nuclei bluish black. Now तीसरा is MTS, Mason's MA trichrome stain. Now this is to identify muscle which is stained red, epithelium which is stained red again, and collagen fiber is stained blue. Now you'll see three colors here, and nucleus is bluish black. तो हो गया ना. Now muscle and epithelium red, collagen fiber blue, nucleus bluish black. Mason's trichrome. Let me show it to you. This is Mason's trichrome. Nucleus is bluish black, collagen fiber blue, epithelium and all the cells red. Okay, now one more stain. Pass. Per-iodic acid skiff. Stain. This stains aldehydic group magenta color. Now what is magenta color? It is sort of like यूसिन के जैसा ही थोड़ा थोड़ा दिखता है, but it is much darker. And this is used for identification of fungal infection. Everything is stained blue except basement membrane, which is stained pink. Now something about plasma cell. It is a defense cell which is responsible for formation of immunoglobulin. It is oval, has circular, eccentrically placed nucleus, which means it is like this. Oval है ना, the nucleus will be placed right here. Don't look at the color, means just ignore. Just shape of nucleus and position बता रहा हूँ. Now, giant cell. This large cell containing multiple nuclei, and it is formed by the fusion of multiple cells. Now there can be two types of giant cells. One is physiologic and another is pathologic. मतलब टाइप तो नहीं है, बट फिजियोलॉजिक मतलब जो नॉर्मल, पैथोलॉजिक जो अब नॉर्मल है ना. ना फिजियोलॉजिक में ऑस्टियोक्लास्ट, सीमेंटोक्लास्ट एंड ओडोंटोक्लास्ट. ऑस्टियोक्लास्ट मतलब जो बोन में है ना. ऑस्टियो मीन्स बोन्स, सीमेंटो मीन्स सीमेंटम, जो सीमेंटम ऑन द आउटर कवरिंग ऑफ रूट उसमें है. एंड ओडोंटो मीन्स टूथ. एंड इसके भी तीन हैं, लैंगरहंस ड्यूरिंग ट्यूबरकुलोसिस, ट्यूमर, giant and Reed Steinberg. This also has two types, binucleated and non-multinucleated. Binucleated has oval eye appearance. Okay, now don't know if I've told about the measurements of tooth, but we'll do that in the next class. Okay, next revision session, not class, revision session. Okay, bye bye, thank you.